Welcome to Didactic. Today is Monday, January 5th, 2026, and I am Dr. O'Leary, a psychiatrist and host of this podcast. This podcast is about psychiatry and neuroscience, and occasionally other things like game theory, or ultrasound, or mental health policy. Today, I'm going to continue talking about what happens when we apply electromagnetic energy to our heads. I'm not talking about ECT, or electroconvulsive therapy. I'm also not talking about TMS, or transcranial magnetic stimulation. I am talking about transcranial direct current stimulation, which is basically plugging your head into a battery and adjusting the amperage. Sound fun? When I was a kid, I remember touching the poles of a 9-volt battery with my tongue and feeling the tingling, kind of stinging sensation. I was also dumb enough once to let a friend fool me into grabbing 2 ends of wires each attached to an electrode on a 9-volt battery and getting a little shock. Fortunately, direct current is generally far safer than alternating current, which is what you get if you plug directly into an outlet in your house. So grabbing those wires didn't kill me. Doing that with an outlet could easily have killed me. Today I'm going to talk about transcranial direct current stimulation because the FDA just approved a device that you can wear on your head at home that delivers 2 milliamps of current in hopes that it lifts the black mood, the melancholy, more recently known as major depressive disorder. I need to warn you that my opinions here are my own and no one else's. Please do not confuse them with those of the federal government, the Department of Defense, the Defense Health Agency, or the Montpellier Vitalist Society. I also use AI to assist me with my research and the content creation, but in the end, what I give you here is my responsibility, and it's my fault if it's wrong or biased. Though I do a lot of fact-checking and try to be fair. If you want to give me some feedback, you can go to scidactic.com and fill out a form there. In any case, I'm going to stop jabbering now and jump right into the content. On December 8th, 2025, the US FDA granted authorization for the Flow F100, a wearable headset designed to treat major depressive disorder in the comfort of a patient's home. This approval is a victory for transcranial direct current stimulation, and one of the first machines that could potentially challenge pharmacotherapies like selective serotonin reuptake inhibitors as the primary first-line intervention for mood disorders. You can take a pill in your home, or you can put a device on your head. For decades, the standard of care has revolved around the modulation of neurotransmitters like serotonin, norepinephrine, and dopamine through chemical agents. Pills we take that get into our body, they affect everything, but also the brain, and they're effective. While these agents are effective for millions, many patients fail to achieve remission with their first or even second antidepressant medicine. And a significant portion of patients discontinue treatment because of side effects, ranging from things like weight gain to sexual dysfunction or emotional blunting. The FL100 uses direct current stimulation, which is basically the same thing as hooking up a battery to your forehead and plugging it into your scalp. The electricity acts locally rather than systemically, which means that it doesn't affect anywhere else on your body but the places you put it and a little bit down below the skull. For depression, it targets the dorsolateral prefrontal cortex with a weak direct current to hopefully change the excitability of the neurons in the brain just below the skull where the electrodes are placed. To fully appreciate the significance of transcranial direct current stimulation, I'm going to call it tDCS. Let's rewind the clock a little bit and talk about other kinds of electricity used by doctors. Adding electricity to the human brain is not even a modern invention. Its practice predates the discovery of the neuron, the light bulb, and even the battery itself. So the earliest recorded instances of electrotherapies date back to over 3,000 years BCE. Egyptians likely applied electric catfish to the body to relieve pain. This practice of using electric marine life, such as electric stingrays, was documented by the ancient Greeks as well. Scribonius Largus is the court physician to the Roman Emperor Claudius, documented a peculiar treatment for intractable headaches and even for gout. It was the application of a live torpedo fish, which is an electric ray, to the affected area. Largus noted that when the fish was placed on the forehead of a patient suffering from a headache, the resulting numbness, a phenomenon we now recognize as stimulation-induced analgesia, could provide lasting relief. Later, the idea that electricity was a primary component of life became popular, especially during the Enlightenment. So in the late 18th century, Italian physician Luigi Galvani conducted his famous experiments on frogs. So he connected the sciatic nerve of dissected frogs to a metal conductor during a lightning storm, and this induced muscle contractions. He concluded that biological tissues contained intrinsic animal electricity. It's basically a vital force that governs movement and sensations. Before this, Descartes thought that the nervous system was basically a hydraulic, because that was the technology he could imagine at the time. Now that experiments with electricity could be done, people started to try to explain movements and motivation as being electric instead of hydraulic. Galvani's contemporary and also his rival, Alessandro Volta, argued that electricity was not intrinsic to the animal, but it was generated by the contact of dissimilar metals. So this led Volta to invent the voltaic pile, basically the world's first battery. And then Galvani's nephew, Giovanni Aldini, in 1801 began treating patients suffering from melancholia, which is basically just a historical name for what we would now recognize as major depression. And he did this using his uncle's principles and Volta's battery. So Aldini applied direct currents to the scalps of patients, and he reported that this could lift the black mood of the afflicted. He also liked to get attention by stimulating the corpses of executed criminals in London, causing their eyes to open and their jaws to clench, much to the horror and also the fascination of the public. And it wasn't long after that Mary Shelley wrote Frankenstein, using the idea that electricity provides a vital force to material tissues. Despite Aldini's early rabble-rousing, the use of electricity in medicine fell into disrepute during the 19th century. It was often associated with quackery. Medical galvanism devices promised to cure pretty much everything from baldness to impotence. But it wasn't until the 1930s that electricity returned to the mainstream. In 1938, Italian neurologists Ugo Cerletti and Lucio Bini introduced electroconvulsive therapy. If you are interested in learning about this process in more detail, my first 3 full episodes of this podcast deal with electroconvulsive therapy. Unlike the subtle modulation of Aldini's direct currents, ECT utilized alternating currents at high intensities to intentionally induce a grand mal seizure. The therapy was incredibly effective for catatonic depression, for mania, and for psychosis that was caused by mood disorders. It wasn't the electricity itself, however, but its ability to produce the seizure that was determined to be the therapeutic agent. The electricity was just merely the trigger that induced the seizure. In its early days, ECT was administered without anesthesia or any sorts of muscle relaxants, so this led to bone fractures and violent convulsions. It also caused significant memory loss and a lot of confusion in patients. But modern anesthesia and better control of the current and the electrode has made ECT far more safe. But culturally, ECT later became synonymous with shock therapy. It was a stigma reinforced by media portrayals like One Flew Over the Cuckoo's Nest, but also reinforced by the fact that ECT was vastly overused for all sorts of conditions, from like bedwetting to trying to cure homosexuality when it first became popular. The reintroduction of more gentle forms of electrical stimulation began in the 1960s. So researchers in animal physiology observed that you could apply weak direct currents to the cerebral cortex of rats, and that could produce long-lasting changes in neuronal firing rates. So these currents were too weak to actually trigger the neuron to fire, but they made it easier for the neuron to fire. They altered the resting membrane potential of the neurons. When a positive anode was applied, it was found to depolarize the membrane, so that brought it closer to firing, made it easier for the neuron to fire. The cathode, or the negative stimulation, was found to hyperpolarize the membranes of neurons, so it made it more difficult for them to fire, given the normal stimulation that they would get. But in the last 30 years or so, direct current stimulation and transcranial magnetic stimulation have been researched extensively. So magnetic stimulation uses changing magnetic fields to stimulate neurons, and I also did a series of episodes on this, if you want more information on that. But it was also shown that applying positive direct current stimulation could make it easier for TMS to make those neurons fire. It was shown that direct current stimulation can induce a kind of neuroplasticity. We call it long-term potentiation in the human brain. And it did this non-invasively without the need for any sort of sophisticated magnetic field generator like TMS requires. Now, unlike ECT, there's really no need to cause a seizure, and there's no loss of consciousness in the patient. There's just stimulation of a certain part of their brain for a certain period of time. The subject generally feels only a mild tingling or maybe a stinging sensation, and they might get a rash on the skin. So TMS and tDCS made it possible to affect the brain without shocking it, but just kind of nudging it. It established the biological plausibility that transcranial direct current stimulation, tDCS, could be a treatment for conditions that were characterized by cortical hypoactivity, such as hypoactivity in the left prefrontal cortex, which has been demonstrated in depression many times. So all of this laid the scientific bedrock upon which the company Flow Neuroscience would eventually build the FL100. Flow Neuroscience was established in 2016 in Sweden, and it was founded by Daniel Manson and Erik Ren. They made it their goal to transition tDCS from a laboratory curiosity to a regulated home use product. Daniel Manson was a clinical psychologist who treated patients with major depressive disorders, so he was familiar with the limitations of the standard care that we give today. Psychotherapy is very effective, but it can be scarce in many areas, and it can be slow to work. It might take many, many weeks. If you're doing psychodynamic psychotherapy, it might take years. And pharmacotherapies, giving medicines, is easier to access, but it's often accompanied by side effects like weight gain or sexual dysfunction. Many patients are also just too depressed to really engage fully in therapy, and they're unable to tolerate the side effects of medicines. TMS and ECT require patients to make multiple trips to the office, and this is often multiple times a week. So Manson thought, we need something that is effective, that can be done at home. Erik Ren, the other co-founder, was the one who brought in all the technical expertise. So he's a neuroscientist and an electrical engineer with a background in artificial intelligence and in computational biology at the Royal Institute of Technology in Stockholm. So Ren was able to miniaturize the bulky systems that were out there before that gave tDCS. And then he helped to make the system more foolproof. That is, one that could ensure that it was used correctly at home without the need for a technician to be present. Before I explain how he did that, I want to talk a little bit about what the FL100 was designed to do. And this is grounded in something called the asymmetry hypothesis of depression. Decades of EEG and fMRI studies have shown that, for some reason, major depressive disorder is characterized by a specific imbalance in the frontal lobes. So in the left dorsolateral prefrontal cortex, there is hypoactivity. And this region is critical for executive functions, emotional regulation, and what we call approach behaviors, to actually decide to go do things. But in depressed patients, it's often metabolically underactive. And this might look like apathy or cognitive fog, and we're having a low mood. So because the left dorsolateral prefrontal cortex is not as active, you may be less motivated in general. There's also hyperactivity in the right dorsolateral prefrontal cortex in many patients. So this region is often associated with what we call withdrawal behaviors, to not do things, to escape. And it's also associated with low mood or negative affect, and often with a lot of anxiety. So overactivity here is linked to things like rumination, where you just sit around and think about how bad things are, and an inability to disengage the negative thoughts that often come around when you're depressed. So there's frequently hypoactivity in the left dorsolateral prefrontal cortex and hyperactivity in the right dorsolateral prefrontal cortex. Remember when I said earlier that an anode can increase the firing rate by depolarizing the membrane, and a cathode can decrease the firing rate by hyperpolarizing the membrane. So if you place an anode over the left dorsolateral prefrontal cortex, it's delivering excitatory current, encouraging neurons to depolarize, potentially fixing the hypoactivity of the left dorsolateral prefrontal cortex. And if you place the cathode over the right dorsolateral prefrontal cortex, it delivers inhibitory current. And this is thought to hyperpolarize the neurons and dampen down that overactivity that has been demonstrated in the right dorsolateral prefrontal cortex. Now we know that it can do this, but whether or not that is enough to be effective to treat depression is the question here. One of the things that made this device able to be used at home is that it makes sure that you don't give too much energy when you're doing it. For example, the device won't even start unless it detects a solid electrical connection with the skin. So this prevents things like hot spots where it heats up part of the skin because there is impedance or resistance there. So it connects via Bluetooth to a smartphone application, and this application serves multiple functions. First of all, for safety and compliance, the app controls the stimulation. A user cannot simply put on the headset, turn it on, and determine what stimulation they want. The app restricts the usage to a prescribed protocol, and that is 1 session per day to get started. And then it prevents overdosing, basically, or misuse of the device. Another thing that the app does is it gives guidance to the user. It teaches the user how to place the electrodes properly and ensure that the headset is positioned correctly over the head so that you actually get the stimulation in the right places. The app also delivers some behavioral therapy modules while you're using it. So these are kind of based on standard CBT principles and focus on 4 pillars, things like sleep, nutrition, exercise, and meditation. So this is kind of a combinatorial approach. There is the stimulation, but then there's also the therapy that they get while they're stimulating. So either of those things could be having an effect, and it could be hard to actually tease apart which of these is working or not. The device, at least in the US markets, has a clinician dashboard as well, so that just allows the clinician to see that the patient is actually using it, and it allows the clinician to see the patient's self-reported symptom scores. So they're filling out scores on things like the PHQ-9 or the MATTRS, and these are tracking depression symptoms. So they could potentially adjust the protocol if necessary. They could say, hey, you know what, let's extend another week on the everyday protocol. Or they could see that a patient is getting worse again for a full 2 weeks, and they could say, hey, let's go back to the once a day thing instead of let's doing this every other day, or however it is. So it gives the clinician a way to actually track progress and maybe adjust some of the dose that the patient gets. Now, if you have a device like the FL100, it doesn't get approved by the FDA the same way that drugs get approved. The drug approval pathway is far more rigorous, but the FL100 had to go through the most rigorous device approval, and that's called a Class 3 approval from the FDA. So they have three classes basically. Class 1 is very low risk, like dental floss. And then class 2 has a moderate risk. So these could be things like condoms or some kinds of medical software. Or it could be just simply making a device that is substantially equivalent to a product that's already on the market. You can see by the way that the device is designed that it's going to do the same thing as something that was already approved through the class 3 novel device or high risk device system, and so it gets approved without having to go through the same class 3. Class 3, though, I mean, even if you're not novel, but you're high risk, things like implantable pacemakers or heart valves will have to go through class 3 approval no matter what. And this is designed to prevent devices that could harm you. And so these devices require pre-market approval. Because the Flow F100 was the first at-home tDCS device indicated for depression, there was no predicate device to which it could claim equivalence, so it couldn't go through a class 2 pathway. It had to go through a class 3 pathway. But one of the ways it got through that pathway so quickly was that it got breakthrough device designation, and that was granted in May of 2022. So the FDA reserves breakthrough device designation for things that meet two criteria. So there's life-threatening or debilitating conditions, and major depressive disorder is definitely a debilitating condition, so it met that one. And then also if there's an unmet need or it gives some sort of advantage that wasn't there before. So unlike antidepressants, which flood the entire body with substances that can have side effects, transcranial direct current stimulation is not systemic, so it has an advantage. It affects the brain, but it spares the liver, the gut, the kidneys, the reproductive system, and all the other organs in the body. Additionally, it addresses an access to care barrier because you can do it at home, and there's not that many things that can be done at home besides take drugs to treat depression. So it had an unmet clinical need and presented an advantage. So that's why it got breakthrough device designation. Now, this designation doesn't lower the standard of evidence that it needs. All of the class 3 devices have the same standard of evidence. It just makes sure that the FDA prioritizes it. For example, as the company is designing its trial, the FDA will work with them to pre-approve their trial instead of wait until the trial is done and then you submit it to the FDA and then they say, well, why didn't you do this? Now go back and do that. So it makes the approval process faster. The FDA works more intimately with the company if they have breakthrough device designation. Now, one of the ways that devices are different than drugs, I mentioned earlier, is drugs require a much more substantial approval pathway. So you have to test the drugs out, you have to show all of their safety, and you have to do some of this with machines too, but it's more rigorous with drugs. For example, the standard for drugs that they're effective is substantial evidence of efficacy. So this almost always requires at least 2 independent randomized phase 3 trials, and each trial needs to have more than 500 patients, basically, to prove that the result is not just a statistical fluke. So the FDA assumes that biology is messy and it requires replication. Even if you have a large number of people in your study, you know what? You got to replicate it. But for devices, the standard is only reasonable assurance of safety and effectiveness. So the FDA acknowledges that device trials are harder to run. For example, blinding is difficult, prototypes are really expensive to make, and if you can show that the device is not likely to cause severe harm, then they will accept one robust pivotal trial with not nearly as many patients in it. So the Empower trial that I'm going to talk about, which purported to show the efficacy of the FL100, only had 174 patients in it. But I'll get to that in a minute. Another thing that's difficult between drugs and devices is drugs, it's usually easier to give a placebo. So you can give a sugar pill with a drug. Now, for some drugs, they have a substantial effect on a patient. They can tell that they took them because of the effect, and so it's hard to blind them. But in general, it's pretty easy to blind a patient that they took the placebo versus the regular drug. There's no way for them to know. For devices, it's often not that easy. Transcranial direct current stimulation causes skin sensations. And so a sham device that does absolutely nothing will not cause any sensations, at least not the same sensations that the device that uses electric currents is going to cause. And so this can result in the end, you might call it a moderate level of uncertainty regarding the unblinding of device trials. And I'll get to that in the Empower study. So to summarize this last part, the FL100 was approved via Class 3 approval, required a smaller study than drugs would have required, and there was a lot more uncertainty about blinding than there would be for something like a new antidepressant. However, it's pretty easy to establish that a machine is safe, a device is safe, and it's more difficult to establish that a drug is safe because drugs might have rare side effects, and so you have to give them to lots and lots and lots of people. Whereas most devices, even if they have some sort of rare problem, it's not going to be that severe. At least not one that's attached to the outside of your body. So the valid scientific evidence that convinced the FDA to give the approval for the FL100 came from the Empower study. The Empower study was cool because it was very decentralized. Participants didn't have to visit a clinic for their daily treatments. Instead, they just were mailed the device at home, and there was supervision provided via video calls. So people from all over the place could participate in this study, and there weren't the same kind of barriers as there are to a study in which you are going in, say, to get an infusion or something like that. So it was much more similar to how you could potentially do a drug trial via telehealth. But with a drug trial, you can't actually show that your patient is taking the medication. And with this machine, you could show that they were actually using it because the program that ran the machine would tell you that. So the sample size was 174 adults, and they had to have moderate to severe major depressive disorder. And this was defined as having a Hamilton Depression Rating Scale score of about 19 or more. And then the patients were randomized to receive either the active treatment or the sham treatment. There were different phases in the study. For the first 3 weeks, they had to do 5 sessions per week, followed by what was called a strengthening phase, and that was 3 sessions for 7 weeks, and this was a total of 10 weeks. And patients were allowed to stay on any stable dose of antidepressants that they were already on. So this actually helps to make the results applicable to the real-world population, say people who partially responded to an antidepressant. They got a little bit better, but they still have a lot of issues. So the machine could be seen to be relevant for that population as well. The difficulty here was having the active sham component, right? So it's difficult to fake giving electrical currents to the scalp. The active group received 2.0 milliamps of continuous current for 30 minutes. The sham group received a device that would ramp up to 1 milliamp over 30 seconds. So this was enough to cause a tingling or an itching sensation on the scalp. And then it would ramp back down to 0 and remain off for 29 minutes. And then it would ramp up again briefly at the end. And this was all to mimic a sensation of, well, it's turning on, I can feel it turning on, and it's turning off, I can feel it turning off. And this may help blinding. So the patient might think they got the real treatment because they could feel something happening, but it wasn't enough of something that it would actually cause any changes to the brain. So that was the thought. It was basically aimed to fool the sensory nerves of the scalp. But there was some difficulty still with blinding. Among the active group, they asked them, couldn't you guess whether or not you were in the active group? And about 77 to 78% of them guessed correctly that they were in the active treatment. In the sham group, only 60% guessed that they were in the active group. That's interesting, especially because it shows that there was quite a bit more uncertainty in the sham group as to whether or not they got the active treatment. And about 40% of them were pretty sure they didn't. So that can affect the outcomes. And this is one of the reasons why in the end, the FDA thought there was enough ambiguity in the blinding that they used the term moderate uncertainty in its decision, which means that we're letting this thing go out, but it might not actually work. There's enough uncertainty we should mention it. The Empower study also shows what I would like to call some statistical tension. So on the one hand, the device appeared to be highly effective, but on the other hand, the clinical significance of the findings is kind of questionable. So let's start with the questionable part, and then we'll move into the apparently highly effective part. The primary endpoint was the change in the Hamilton Depression Rating Scale at week 10. The active group improved by 9.4 points. The sham group improved by 7.1 points. So the difference was about 2.3 points. If you want to get more specific, it was 2.27, but we don't need to go that far. While statistically significant, the difference was definitely statistically significant. The 2.3 difference is modest at best. To put this into perspective, a lot of people argue that having a 3 to 5 point difference is what you would need to be noticeable by a patient. So we call this clinical significance, and I'm going to get back to that in a minute. But let's move on to what appeared to be the very large improvements people had. There were some binary outcomes here. There was something called the response rate and the remission rate. So you either were a responder or you were not, or you remitted and you were not. The response rate was defined as greater than 50% improvement in your score. If you had a score of 30, you would respond if your next score was better than 15. Now, to remit meant that you were basically symptom-free, and symptom-free was considered a score of 7 or less. There were also other measures used, but I don't have time to get into all of those. I'm just going to be talking about the primary outcomes here. So it looks very impressive if you consider the response rates and the remission rates. For example, in the active group, the response rate was 58.3%. About 60% of people responded, but in the sham group, it was only 37.8%. So about 40% of people responded. So that's about a 20% difference. It's pretty big. And it was very statistically significant. The remission rate as well. The active group had a 44.9% remission rate, so about 45%. And the sham group had a 21.8% remission rate, so about 22%, about a 23% difference. So if you think about this in terms of the number needed to treat, that's basically, you take the statistical percentage difference between the two and you invert it. So for 20 over 100, for a 20% difference, if you invert that, you get 5. So what that means is you would only have to treat 5 people with the real thing versus the sham thing to get an actual response. You only have to treat 5 people to know that you have really treated someone. In the world of antidepressants, numbers needed to treat are often around 2 to 10. So a number needed to treat of 5 is considered pretty excellent. Also, it was very safe and tolerable. There were no adverse events. Common side effects were really just dermatological, some skin redness and rashes and irritation, but nothing big. There were 2 patients, though, who did get burns. I mentioned earlier that this machine could burn you, but when they looked into it, they found out that the patients weren't following the instructions, and instead of using fresh pads that were wet with a saline solution, they used dry pads. This created a lot of impedance or resistance to flow, and that made it heat up. So they didn't follow the instructions. That's why they burned themselves. But they also noticed that, well, some people, about 1%, more than 1%, burn themselves by not following the instructions. Now let's look at what seems to be a big difference between the very modest improvement in the actual overall scores, but a huge difference in response rate and remission rate. So what's going on here? We know that the results of this study are probably reliable because the p-values were very good. The study was well designed. So it wasn't as if we're thinking, oh, this study has a huge amount of bias in it and we can't trust the outcomes. So it's reliable, but what is the utility of the outcomes is the second thing to ask. So there's this concept called the minimal clinical important difference. And this can be thought of as clinical significance instead of statistical significance. Is anyone really going to notice a difference between faking it and actually doing it? Or are they, for all practical purposes, the same, even though the numbers are different and you get a statistically significant difference? Think of this in terms of like a weight loss pill, and the people who get the pill lose 10.5 pounds. The people who get the fake pill lose 10 pounds. And this is over the course of like 6 months. Like, who's really going to notice the difference there? Are you going to take the chance of taking the pill that might have some side effects, even though it wasn't any different than taking a placebo in a real way? The numbers were different and they were statistically significant, but nobody really noticed the difference. That's what I'm talking about here. That 2.3 difference on a scale is probably not noticeable to most patients because the minimally clinical important difference on the Hamilton Depression Rating Scale is generally thought to be at least 3 to 5 points or more. But if that's the case, how do we explain the huge difference in the remission rates and the response rates? The difference can be explained by what's called the cliff effect. A whole bunch of people are huddled up by the edge of a cliff. Some of them fall off the edge, some remain there. The cliff is straight up and down, like you're either falling off the cliff or you're not. There's no sliding down the hill. Depression scores in a population generally follow a bell-shaped curve, so there's a nice hill by which you could go up and down the curve. This is also called a Gaussian distribution. Now, because the curve is like it is, there are a lot more values near the center of the curve. That's where most people are. Most people are close to average. There are a lot fewer values near the edge of the curves, so most people are not on the fringes. The threshold for remission, if you define it as a score of less than 7, cuts through one of the densely populated areas of that curve. So a small shift on the curve pushes many more people over an arbitrarily drawn line than it would if that line were drawn near the fringes of the curve. Even though there were only a couple points difference, that caused a lot of people to be classified as having remitted. But is there any difference between the remitted people and the people who responded? Clinically, they didn't ask that. If you have only 2 possibilities and nothing in the middle, you can have a huge looking difference when there really isn't one. If you've been listening to this podcast before, you know that I often bring up something called Bayesian statistics, and I really kind of drive it into the ground because I think that a Bayesian framework is way better than the way that we do things now. And so I'm going to explain to you a little bit about why, if you take a Bayesian perspective on this study, you might come to a very, very different conclusion than did the study itself. And you might understand why the FDA wrote down there's a moderate level of uncertainty here as to whether or not this thing's going to actually work. But it's safe, so let's give it a try. So there's basically 2 kinds of philosophies on probability. There are more, but the 2 big ones are frequentists and Bayesians. So a frequentist basically thinks something like this. The probability is the frequency or the number of times an event occurs if you were to be able to do that event over infinite number of trials. It assumes that the parameter that you're estimating is fixed in the population. It doesn't change, and the data that you're getting from the population have been randomly sampled from that population. And it basically says, how likely is this data given the population you're taking it from? Frequentists don't actually know the population, they're just assuming it. And they make lots of assumptions. For example, that the population has a certain distribution, and that the data that you got from the population was randomly drawn from it, like there's no bias at all. Bayesians don't think like that. They just assume that there's all kinds of things contributing to why you got the numbers. So instead of thinking about things as being either true or false, they have a probability that something is true, or a degree of belief, or you might call it a confidence level that something is true. So instead of Bayesians thinking that the population you observed it from is fixed, they only think that the data you got is fixed. That's the only thing we can say is real, is the data, what we observed. The parameter itself is actually a distribution of probabilities. Now, that sounds kind of weird. Let me simplify it by saying this. A Bayesian would say, after they read a study, well, given this data, what's the actual probability that the drug works? A frequentist would say, well, the probability is the data itself, but the Bayesian would say, well, what have similar people in the past gotten when they've done a similar thing? So you can't just consider the data right now, you have to consider the data in a huge context. That's what Bayesians do. They add way more context to everything. And they can be really annoying, like me. So I'm not going to come in here and tell you that the FL100 works. I'm going to tell you that there's some data that it does, but whether or not it does, I don't know. That's uncertain. So here's how you might think of the data a little bit more like a Bayesian would. A Bayesian has a kind of prior probability going into anything. That means that like going into something, they're like, I think this will probably be this way, or I think it'll probably be another way. For example, like for transcranial direct current stimulation, the evidence is mixed, but there's a lot of evidence that shows that it doesn't work, actually. So prior studies that were similar to this one and found no evidence for effectiveness would affect the way that you think about this. And there are studies out there very similar to this one that found absolutely no effect. I will provide links to those in the transcript at didactic.buzzsprout.com. And there's also studies that show that transcranial direct current stimulation has little to no effect on healthy adults for a range of different cognitive measures, like their working memory and their ability to perform different tasks. So really, there's not a lot of evidence that ahead of time that makes me suspect that this device even should work, and there's quite a bit of evidence that makes me think that it won't. So I have a skeptical prior. Coming into this, even if they get a result, I have to still be skeptical of it because I have to multiply the likelihood of their result by the prior probability that they would even have got that result in the first place, which for me is low. There also has never been a dose response curve established for transcranial direct current stimulation for depression in the past. And even in this study, if you look, they had no dose response in this study. They didn't have a big enough sample, if there was one, given their data, to show that, but they couldn't find one. And if there's no dose response, that means that it's less likely. Does the brain really just fall off a cliff at some point and now you're better? Or is there a dose response? Usually there's a dose response, but there's not one in tDCS. So that lowers the posterior probability that this actually is an effective treatment. Medicines have dose responses. This has never demonstrated one. So if we feed these skeptical priors into the whole thing and we get the likelihood of this data, we can also take the data itself and say, well, what do I know about the data that might make me suspect the data? And one of the big things we know about the data, a lot of people seem to know they weren't getting the real thing, and that's going to affect whether or not they get better. You already have crippled data, and then you have crippled priors, and you have this cliff effect, and you have a very small, modest potential improvement. To me, that all adds up to, I don't think this probably helps. My level of uncertainty is more than moderate. My level of uncertainty is pretty high. Let me summarize there what I just did to you, talking about Bayesian versus frequentist stuff. So if I were a frequentist and I were thinking about the probability that transcranial direct current stimulation is actually better than faking it, I would look at the p-value of 0.012 2, and I would think to myself that, wow, it's pretty highly likely that this device actually had an effect. And that effect is probably somewhere within the 95% confidence interval that was calculated by the study. But a Bayesian would say, bull malarkey. I've got to adjust for my priors, and I've got to adjust for the unblinding bias. So a Bayesian would treat the unblinding guess as a variable and modify the data itself to modify the likelihood of that data. So they would model the correlation between guessing active and guessing sham and actually getting those things. They would take the cleaned up likelihood and they would adjust it by applying the priors. How much confidence did I have ahead of time? If the confidence was like pretty low, let's say 0.25, 25% ahead of time that I thought this would actually work out, I could multiply that by the likelihood. And if the likelihood was already a little bit near 50-50, then here I am still with low confidence that this is actually effective. That's where Dr. O'Leary comes out. I would really appreciate it if you could read the study and maybe apply some Bayesian thinking to that study, and then send me your reasoning and we can kind of argue it out, because I think there's room for a lot of people's opinions here, not just mine. If you want to send me your opinion, you can send it to info@sidactic.com, or you can put a comment on the YouTube video that comes out for this podcast, or you can fill out a form at sidactic.com and let me know what you think. If the FL100 is actually as effective as some people think it is going to have a huge impact on the way that we treat depression. It's probably going to have a huge impact on the way we treat depression anyway, but whether that impact lasts, whether we keep using it, is a different story. If it's truly effective, we will probably keep using it. If it's no more effective than anything else, it'll probably fade away. And then there's this one last thing, because I mentioned it earlier, that complicates the picture. They're using an app to give cognitive behavioral therapy to someone while they're stimulating their brain. So how would we know whether or not any effect this machine had was due to the therapy itself or due to the effects of the machine? Good luck with that question. Thank you for hanging out with me today. I am Dr. O'Leary, and this has been an episode of Sidactic.